So I can record this. All right. So hello everyone. My name is Hryhory Sinenka. I'm doing a PhD in Strasbourg at Strasbourg University within this Allostery in drug discovery Marie Curie network. I will talk about that in a few moments. And currently, what I'm doing is this meeting called Science Café, which is one of the formats recommended by our Marie Curie network to engage, especially with general public, to explain the PhD projects which the early stage researchers are working on. So feel free to ask me any questions after the presentation, and even during the presentation, you can unmute yourself and without any problem interrupt me, because the main idea of this Science Cafe is that you understand everything what I'm talking about, and this presentation is not focused on professionals, so it is focused on general public. So at the same time, if you are a scientist, if you have scientific background, you maybe should not expect some very deep technical dive in, but we can of course talk later with you one-on-one if you're interested in the topic which I will be covering. So with that said, let us start. I'm very thankful for everyone who is present. So our Marie Curie network is called Allostery in drug discovery. And according to Jacques Monod, who is a very famous scientist in, especially in synaptic receptors field, allostery is the second secret of life. And if we overview the ALLODD project, and ALLODD stays for Allostery and Drug Discovery. So ALLODD project is a collaboration between 24 academic and industrial organizations, and this project is focused on training 14 early stage researchers. So early stage researchers are PhD students who are assigned to a certain country, to a certain university, where they are working for 3 years to defend their PhD thesis. And this ALLODD network is sponsored by Marie Curie Action. So this is a European Union funding. I guess one of the most well-known ones, where the budget from the European Union is gathered and spent on different scientific projects of significant need. And ALLODD is focused on training, first of all, on training a new generation of scientists, uh, especially to explore the concept of allostery in drug design. And I will talk about that a little bit later. And of course, uh, since it is a drug discovery project, the focus is to develop techniques, to develop an approaches, to ideally find new molecules, new compounds which can regulate the function of various proteins. So you can see in here the various links to ALLODD website, to Twitter, to YouTube, to LinkedIn. Please feel free to join. Uh, and if we look at the ALLODD beneficiaries and partner organizations, so beneficiaries are first of all those where the PhD students are working in. So, for example, I am working at the University of Strasbourg. Some people are assigned to industrial bodies like Merck, like Johnson Johnson. Some people are in other universities, like University of Barcelona or Karolinska Institute, and so on. And there are also partner organizations. These are organizations who have either sponsored the project or sometimes which suggest the secondments. So, for example, in my case, and I am early stage researcher 11, in my case, the mobility was done at the University of Geneva uh, for 2 months, also at the Forschungszentrum Jülich, which this is a research centrum Jülich in Germany. And also I am planning to have a secondment at Johnson& Johnson towards the end of my thesis, which is relatively soon because I will defend this September. And so let's talk about the allostery. So what is that? It is known that most current drugs are designed to bind directly to the primary active sites of their biological targets. However, allosteric modulators uh, are binding to alternative binding sites. And these alternative binding sites may be much more accessible to, especially to develop novel medicines which are not discovered yet, compared to well-known primary active sites. But this is the main idea of allostery. So whenever you have a protein which corresponds to maybe some kind of disease, especially its malfunction, and you want to bind a molecule to this protein to change its behavior. So you have very often two ways. You can bind it to primary binding site, to main binding site, very well known, or you can bind it to allosteric binding site, which is less well known, which is an alternative one, but which still can produce some response from the protein, and in this and in this way we can try to find much more novel medicines than are available on the market right now. And so what we should also note is that allostery as a concept is very much underexplored, and only now the scientific community starts to focus on that much more, because it seems like the classic drug discovery, which is focused on main binding sites, uh, sometimes comes to a brick wall. So sometimes it identifies many proteins as undruggable. And so allostery is the concept which can solve this issue and open up for medicines for maybe currently incurable diseases. And my project is focused on so-called synaptic receptors. So what are they? For example, one of the examples is glycine receptor, with which I'm working. Mm, these are basically proteins which are inserted in lipid membrane, which is just made of so-called lipids, which are fatty long molecules. And what kind of function do these synaptic receptors, among which glycine receptor is, what kind of function do they address? So in our brain, in general in our body, but especially in our brain, um, there is a massive communication network, which is made of neurons or nerve cells, which send electric signals to each other, and doing that, they control information and information flow through our brain, for example, and our body. But these signals, they cannot just jump directly from one neuron to the next. Because if you look at this picture, we can see that there is a small gap, which is called a synapse. And in this gap, what happens is that the signal transduction happens. So the electrical signal travels across our brain, our neuron system, through this small gap, which is called synapse. And how is it happening? So you can look at this picture. So they are so-called neurotransmitter molecules, which can be very small molecules, which modulate these synaptic receptors. So, for example, this molecule can be glycine. And so if this glycine binds to glycine receptor, what can happen is that uh, glycine receptor opens this channel across the membrane, and through this channel, ions such as calcium, potassium, just ions which can basically conduct ion current, they start to travel from the out of membrane region to the intramembrane region. And this is actually how the signal transaction happens in our organism. So, and in here, this is a much bigger scale of what is happening. So you can see this in red, these small neurotransmitter molecules. And in here, in these sort of green semicircles, the receptors are depicted. And what is going on is that these neurotransmitters, they bind to receptors. Receptors open their channels, and through these channels, ions can flow. So this is basically how in our brain the signals are transferring from one cell to another. And unfortunately, as any kind of protein, these proteins can experience malfunction, so they may not function properly. And the malfunction of synaptic receptors is very often related to many serious diseases, especially of neurological nature. So you can see in here chronic pain. Some people even connect this to Alzheimer's or Parkinson's disease, although of course, in Alzheimer's, the mechanism is very complex. We don't know that for sure. However, so chronic pain is for sure related to malfunction of synaptic proteins. There's strong evidence in favor of that. Parkinson's disease, schizophrenia. So at least an attempt to restore the function of these synaptic receptors can influence much better treatment of at least some of these diseases. And the key here is that we would like to try to control this function of synaptic receptors, to especially to reverse their malfunction in case there is such situation. But how do we do that? And so, as of course, we would like to find new drugs, so-called modulators of synaptic receptors, which can bind to them. And why I mentioned also the concept of allostery is that you see the binding site is outside of this main channel, which I have described in here. So the channel is conducting ions, so this is one thing, but what is happening is that when the molecule binds to a certain place of the protein, this channel can be influenced. So this is not a directly place of the channel itself. But so now the question is, how can we find such compounds? How can we find new drugs, basically? And in here, new technologies come into play. So first of all, what we should do is develop some kind of theory. And we are lucky that the theory of modulation of synaptic receptors is already developed. And it is based on different conformational states of these synaptic receptors. And what I mean by conformational states are basically geometrical states in space of these proteins. For example, as obvious from the biological explanation of their function, they can be closed. So this channel can be closed, the channel through which the ion flow is happening, and this channel can be open, of course. And what is going on here is that drugs can bind to a closed state of the receptor and transform this closed state into the open state, and vice versa. Maybe some drugs can bind to the open state and transform it to the closed state. So it depends on the function of the molecule itself. And the problem here is that the pharmacological properties of any compound, let's say how strong it is as a potential medicine, as a potential drug, how active it is, how stable it is, they are related to both of these states. So basically, according to current theory, one has to understand how strongly the ligand is bound to both of these states separately in order to understand, let's say, whether this medicine will be good or bad, whether it will be strong or weak, whether it will shift the protein state into more open or into more closed. And what can we do in this situation is we can calculate binding energy values of modulators of synaptic receptors, basically of these drugs, to different states of the protein, and then connect them to their pharmacological characteristics. Now, what is interesting is that one cannot do that experimentally. So experimentally, unfortunately, one can only take a given molecule and to measure whether, let's say, it is a strong drug, it is a weak drug, it is active, it is not very active, but one cannot experimentally measure the binding energy values of a given molecule to open or to closed state. But unfortunately, this is what is needed to find new molecule. And the only way, therefore, to do that is to use computational approaches and to use various computational techniques to calculate these these binding energy values. And well, this approach can open up a field of computational neuropharmacology. So what is going on and what is planned in my PhD project is to calculate binding free energy of various molecules to this open state of the protein, to this closed state of the protein, and from there derive whether this protein, whether this given molecule is active or not active, strong or weak, and therefore to prove this theory. So this is very general description of my project, of what I'm working on, and I'm very thankful for your attention. Of course, if we go deeper, it will be much more technical, I think. So if any one of you are scientists, we can talk later on a more precise basis. But I would be very thankful for any general questions. So if you especially didn't understand what I was talking about, because this is my sort of first engagement, I would say, with general public, I am very thankful for any forward questions, and I will be very thankful to hear them and would be glad to answer. So thank you very much, everyone, and let's talk about these general approaches. And please write me in personal mail, in personal messages if you want to discuss something on a more deep scientific level. Thank you very much. All right. If no one has any question, uh,
I have a question. So you want to compute binding free energies between your small molecules that you will be virtually screening in both states, open and close?
right,
That's
right, that's correct.
and do you know if the allosteric pocket is I mean exists in both states as well, and if that pocket has a different shape?
Uh, yes, yes, we know we have cryo-EM structures, so we have experimental structures, and well, so if you're a scientist, I would be very willing to talk on a bigger sort of scale. But yes, to just answer your question directly, we have experimental structures, and uh, this is why it is very much possible.
Okay. I'm a scientist, as you say. I'm a
Yeah.
computational chemist, and I've been
Yes, yes, yes. It's just that
on
this
the
is Science Cafe, so the main idea what I'm obliged to do here is to engage with general public. So the presentation is very much, I would say, basic, and it doesn't go to any technical details, to technical results, for example. And this is, yeah, it's not also so easy to engage with very general public. So not so many people visited this this science cafe, but in general. So my point is that if anyone is an expert here and is really interested, we can even let's say disconnect and then connect once again to talk maybe in a more precise way. Just outside of this sort of science cafe, even maybe immediately after, if you want. I can just stop sort of the demonstration, I can stop the recording and yeah. So I hear I
maybe
also.
other people
Uh-huh.
have questions as well.
Mm-hmm. Yep, for sure. Thank you very much.
Maybe other people here have questions as well.
Maybe. So let us see. Let me check. Um, So there is okay, Ulrich. Uh, I don't know if this is technical or not a question, but for this project, both activator and inhibitor of the protein, of which of these are we focusing on? Oh, thank you very much, Ulrich, for the question. So I am focusing on both well, I would say on both active and inactive ligands. So some of them, let's say, are considered as indeed inactivators. So they sort of shift this equilibrium to more closed states, for example, gamma aminobutyric acid, and some of them are very active, let's say glycine. And the idea is to predict using calculations that, let's say, glycine is a better modulator than this gamma aminobutyric acid, and yes, and vice versa. So yes, we are working basically with both, I would say, inhibitors and activators, and we have experimental data for them both. But thanks a lot for asking. All right. So, in case there are no other questions from the general public, I am very thankful for this Science Cafe. I'm very thankful for everyone who attended and for your questions. So, from that, I would like to stop share.